afternoon, sharks. Today I'll be pitching to you a virus. So, for some context, we have a fungus called powdery mildew, which is the bane of agriculture. It infects crops all over the world, spreads fast, and farmers are currently trying to fight this with fungicides that are expensive, messy, and increasingly ineffective. So what if we found an alternate solution to fight this pest, which reduces crop viability and reduces the amount of food that these plants can produce? So we went hunting in large public datasets using the Serratus tool and found a virus or viral sequences in these datasets of the subspecies of powdery mildew fungi called Golovinomyces, Cichoracearum. So it appeared in 3 datasets. 2 of them were actually from this exact fungus, and one of them was a dataset for tobacco leaves, which we assume is because this tobacco leaf was likely infected by the fungus, because it's very unlikely for a virus to be able to affect both the fungus and the plant. So, how do we know what this virus even is? So the little identifier sequence that we picked up on, we were able to compare it to using a tool called tBlastN to see what other viruses or what other organisms have a similar sequence like that. And the hits were mito-like viruses and mitoviruses. This is our first clue to think, okay, we're very likely picking up on a mitovirus. Now, what is a mitovirus? A mitovirus is a virus that specifically affects the mitochondria of fungi. Now, why is this exciting? Because, as we all know, mitochondria is the powerhouse of the cell, and this means that a virus that is stealing energy from the energy source, this means that it is reducing the energy of the species that it's infecting. And if we're reducing the energy of our fungus in question, then we can reduce its viability and in that way protect our crops. Now, this isn't enough sort of like detail, enough sort of like data analysis to really prove this point. So we dug a little bit deeper and went into the sequencing. So using the sort of the little snippets of sequencing data, we were able to see that and able to find, I guess, a big open reading frame of about 2,000-ish nucleotides that sort of matched up with our sequence of interest. Now, what this means is this 2,000 nucleotide sequence is our basically the protein that our virus is producing. Mitoviruses produce one protein, and it's very good at replicating. We verified this, or we were able to not only like find this reading frame, but find motifs inside of it that match what we expect from a mitovirus. So now we've confirmed that there's a mitovirus living inside this fungus. Like, what does that mean? How can that help us? So papers have come out showing not only can these viruses reduce the viability of the fungus, but they can spread from virus to virus. So the proposition that I have is that we can use this virus as an agricultural tool to help tame the spread of powdery mildew. And because of the scale of agriculture sort of globally or even in certain regions, this can very easily reduce millions of dollars worth of sort of loss of plants and lots of food. And this is using very conservative estimates. So I'm very excited about discovering this virus and what it's able to do.